Okay, guys, so last time in our last video 5, we were talking about carbohydrates, and now we're going to talk about hopefully the rest of your organic molecules. Definitely we will talk about proteins and nucleic acids. We might also fit fats in there, but I have not done a good job of keeping these videos short like I promised y'all I would. So it might just be proteins and nucleic acids. We will see. Here, I'm sharing my screen, and we're talking about proteins. Okay, so proteins, they are one of your organic molecules, and remember that your carbs, proteins, and nucleic acids, those 3 organic molecules are made up of monomers, which we will talk more about with proteins in just a minute. They have very variable structures and functions, more variable structures and functions than any of the other organic molecules. So you've got so many proteins that function to keep you alive and keep your body functioning and working to its best ability. Now you've got lots of different types of proteins, like structural proteins such as collagen, collagen and elastin. So when you get older, right? And like old lady skin, not old lady skin, come on, Eden. Old people skin, because old dudes get old too, right? All right, and your face starts getting like weepy and just not like your skin doesn't bounce back, and that's because of the proteins, because your body will stop producing elastin and collagen. You can supplement, like, kind of, but it's a situation where, like, if you start on these collagen and elastin infused like creams and things, it's going to, you are not going to get back what you once had. You're going to reduce further like stuff to your skin. Does that make sense? Like, it's going to reduce further losses and further weepiness, I guess. Okay, I'm sharing it again. So collagen and elastin, contractile proteins, so in your actual muscles. So these are going to be muscles that you can control. You have particular proteins that are going to enable you to contract those muscles. Got transport proteins that has pores running through them. That way you can move a substance from outside of a cell to inside of a cell, for instance. Storage proteins, enzymes. We will talk about enzymes once we get to energy. Very variable structures and functions. Okay, so the monomers of proteins are amino acids, and all amino acids have the same general structure. All amino acids have the same general structure. All amino acids have the same general structure, and there are 20 different kinds of amino acids. So you are not going to need to memorize these amino acids at all. If you need them, I will give you a chart on your exams, for instance. But your general amino acid structure, I will not ask you for a chemical structure. I will, though, ask you what sorts of groups, what sorts of items are in all amino acids. Okay, so first, you've got an amino group right here, a nitrogen atom and 2 hydrogen atoms attached to the central carbon atom. Amino group, a carboxyl group right here. All amino acids also have this central carbon atom, and then they also have what's called the R group. Now, you've got 20 different types of amino acids, and they all have the amino, carboxyl group, central carbon atom. What makes them unique is the R group. Okay, what makes them unique is the R group. Here's an example of how the R group can differ between different amino acids. So here you've got a really simple R group, like in glycine, you just have a single hydrogen atom making up your R group. And note, the R group, regardless of whether it's super simple, like glycine, or super complex, like tryptophan, the R group is situated in the same spot on the molecule. Okay, so simple hydrogen atom versus cysteine versus a really complicated R group like tryptophan. But notice, still each one of these has the amino group, the carboxyl group, central carbon atom, and the R group. So the R group is the variable region. This is the part here, here, and here. The R group is the part that differs between the 20 different types of amino acids. Now, dehydration synthesis and hydrolysis are the same in carbohydrates as they are in proteins, as they will be in nucleic acids, okay? So here you have two monomers, and remember the monomers of a protein are gonna be your amino acids. So you've got one amino acid here, one amino acid monomer here. If you wanted to join them, you would be going through the process of dehydration synthesis. Make a bond by losing a water molecule. You're forming this bond in proteins is called a peptide bond. You don't have to know about phosphodiester bonds. You don't have to know about these really complex sorts of bonds that other polymers have and other organic molecules have. I don't even just know that the bond between amino acids is called a peptide bond, okay? So dehydration synthesis, you are forming a peptide bond between two monomers with the loss of a single water molecule. Hydrolysis is the opposite reaction. You're taking this dipeptide, this protein polymer, and you want to break it down into the amino acid monomers. You're going to do that by adding in a single molecule of water in order to break the bond between the amino acids. Now, we talked about how proteins have the most variable structures and the most variable functions, okay? You saw some of the functions in the first slide with proteins, and now here is the structure part of it. Proteins have really amazingly complex three-dimensional structures, and to get to that beautiful three-dimensional structure, you have to go through these several different steps. Okay, so first is going to be your primary structure, and in your primary structure, this is just you have an amino acid chain. You have all of your particular amino acids to make that protein strung together in the correct order with peptide bonds linking each of those amino acids. Okay, so basically think of the primary structure as like just the chemical structure, okay? Then you're gonna have this amino acid chain folds and curls itself into alpha helices and beta sheets, okay? Now, if you will remember back from probably chemistry, you talked about this in, you're gonna have some amino acids that are hydrophobic, some amino acids that are hydrophilic. You're gonna have some amino acids that like to be near each other and some that don't. Based on those different affinities for water, for each other, whether they're polar, non-polar, they are gonna fold into alpha helices or beta sheets, okay? So for instance, if you were hydrophobic, do you like water? No. You repel water, okay? You want to steer clear of water. So would it be more helpful for you to be in an alpha helix that has a spot that you can sort of hide from water, or a beta sheet where every last bit of you is exposed to water? These are different types of structures that are going to contribute to the three-dimensional structure, okay? And it's called the secondary structure. Primary structure, you had your amino acid chain. Secondary structure, that amino acid chain folds and curls into alpha helices and beta sheets. Next, you have your tertiary structure. So you have, by the time you get to a tertiary structure, you have a fully functional protein, okay? In order for a protein to be functional, it has to be in its three-dimensional structure. For a protein to be functional, it has to fold. So for instance, is this a fully functional protein? No. No, it's just an amino acid chain. Here, though, this is just a peptide, okay? It's not a fully functional protein yet. Down here, where you have the alpha helices, the beta sheets, everything going on right, it's folded correctly. You've got a three-dimensional structure as your tertiary structure, okay? This is a functional protein. Next, there are a couple of different types of three-dimensional structures, though, because if you can see here, you've got a single like color for this protein versus in your quaternary structure, you've got this lighter purple and this darker purple. And that's showing you that two different peptides. So you had one peptide like this, it folded, became its own protein, and then you had another peptide different from this that folded, also became its own protein, and now they're interacting together to form a novel protein. So tertiary structure, this is when a single peptide has folded into its three-dimensional structure. It is a functional protein. Quaternary structure is where you have multiple peptides making up this single protein. They have interacted together to form something different from just something that has a tertiary structure. Okay, does that make sense? Think of when we talked in chapter 1 about emergent properties. Okay, you can have peptide 1, it folds into its three-dimensional structure, it is functional protein, it is doing its job, and that's great. But you can also take protein 1 and have it interact with protein 2 in order to form an entirely new protein with an entirely new job. Okay, that's why proteins have the most diverse structures, functions, three-dimensional structures. Okay, so you only have a quaternary structure when you have more than one peptide that's making it up, more than one amino acid chain that's making it up. Okay, so in this example, like ignore the side of that, okay? If I gave you this picture on your exam, and you can see that there are a bunch of different colors signifying lots of different peptides that are coming together to form a three-dimensional structure of a protein, a single protein. Is this a tertiary structure or a quaternary structure? It's a quaternary structure because it has these many different peptides interacting together to form this single protein. Okay, if this was all just this gray color, what structure would it be? It would be tertiary. Okay. All right, so now this is an X-ray crystallography image of a protein, and if you think about, like, this might be an enzyme. Think about this being a digestive enzyme. In digestion, there's breaking down molecules, right? So if this is a digestive enzyme, a molecule has to be able to come into this enzyme, sort of sit in here in the active site and be broken down and then released. So this structure is the perfect shape for its function, the perfect shape for its function. Now, if you had something go wrong while you were building this protein and you had a part of this three-dimensional structure that closed this like entrance to this digestive enzyme, would it work anymore? No, because what you'll see over and over again in biology is that structure meets function. Structure meets function. If you have a pore where like something has to sit in order for you to break it down and it's blocked, it's not going to do that job anymore. Think about the neck of a giraffe. Think about like the long proboscis of a moth and butterflies. Like structure meets function. Structure meets function. All right, guys. So we use food as a building material. We consume organic molecules from other organisms. We break those molecules down into smaller components, and then we use it as a building material to build our own body plans and to give us energy. Now, essential nutrients. What do you think an essential nutrient is? In biology, when you have an essential nutrient, this means you need it, it's essential, you need it, but you cannot make it, okay? So it's essential for you to consume something that has essential nutrients, okay? Substances needed by the body that it cannot make itself. So an example of this are essential fatty acids. These are fatty acids we cannot make from simpler molecules, but that we need in order to have healthy functioning. Essential amino acids, we'll get to on the next slide. What is an essential amino acid? Look, an essential amino acid is an amino acid that we need, but we cannot make ourselves, okay? There are 8 essential amino acids. 20 total amino acids, meaning 12 of them we can make ourselves, 8 of them we have to get by consuming other sources, right? You do not need to know what the 8 are. Here are the 8 essential amino acids. I'm not going to ask you to list them. You do not need to know them. You should know that there are 8 of them though. So human cells cannot make them, but we need them to survive. Animal products are complete, meaning if you eat eggs, if you have dairy, if you eat meat, you have all of the essential amino acids that you need in that thing. Plant proteins are incomplete, meaning that, for instance, they're deficient in one or more essential amino acids. So if we're looking at, let's say you were at a 4th of July barbecue and you had corn on the cob and you had baked beans, okay? Your corn, look at this yellow box right here. Your corn has 6 out of the 8 essential amino acids, so it has plenty of them, but does it have all of them? No. Corn, if you only ate corn, you would be missing tryptophan and lysine still, because this is a plant product. Beans are also plant products. Beans have 7 out of the 8 essential amino acids. They are only missing methionine. But if you eat both corn and beans, if you eat different plant products, you're getting all 8 of your essential amino acids. Okay, so what does this mean? Does this mean that people need to eat animal products? No. Does this mean people should not be vegan or vegetarian? No, of course not. But it means if you are vegan or vegetarian, that it's important for you to consume a varied diet. Okay, consume a varied diet. You cannot just eat salads. You can't. If you do eat salads, make sure that a bunch of other stuff is on there. Okay, tomatoes, nuts, like whatever. Okay, so that was proteins. Now we're at nucleic acids. Nucleic acids, you've got DNA molecule, DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. So DNA and RNA are your 2 examples of nucleic acids, and these molecules contain genetic information. Hiccuping. These molecules contain genetic information. So here, you're going to use your DNA to make RNA, and then you're going to use RNA to make your proteins, which we will get into later this semester. The monomers of nucleic acids are nucleotides. Think about DNA. You might know them just from the letters, okay? What are the letters of the nucleotides that you know? A, T, C, and G for DNA. Adenine, thymine, guanine, and cytosine. Now, just like your amino acids, which please, a nucleic acid is an organic molecule. An amino acid is a monomer of a different organic molecule of proteins, okay? So don't confuse those 2. Just like amino acids had your amino group, your carboxyl group, your central carbon atom, and your R group, your nucleotides also have these standard groups, these standard structures, okay? So regardless of whether you are adenine, thymine, guanine, or cytosine, or uracil in RNA, your nucleotides have a phosphate group, a sugar molecule, and a nitrogenous base. Okay, now, what is the difference in the nucleotide structure? Like, which group differs if you have adenine versus thymine versus cytosine or guanine? Does your phosphate group differ? No. Does your sugar molecule differ? No. Your nitrogenous base is going to be the variable region of your nucleotides. But in between your different amino acids, see, I just did it. I hate that when I do it in front of y'all. I hate that. Please don't do what I just did. In between your different types of nucleic acids, your DNA and your RNA, deoxyribonucleic acid versus ribonucleic acid. That's referring to your sugar backbones, okay? So DNA has this deoxyribose ring of sugar backbone versus RNA has just this ribose ring of sugar. We'll get more into that later. So the overall structure of DNA and RNA differ by their sugar molecule. Your nucleotides, the variable region is your nitrogenous base. Dehydration, synthesis, and hydrolysis again. All right, dehydration, synthesis makes it, hydrolysis breaks it. You make a bond with dehydration, synthesis. You break a bond with hydrolysis. So for instance, if you were making a molecule of RNA and you wanted to put adenine, uracil, cytosine, and guanine together, you would need to make bonds in between those particular nucleotides, okay? So here you are, if we're making bonds, you're taking two monomers for nucleic acids. In this case, this is one nucleotide. Here you have a separate nucleotide. Dehydration, synthesis, you are going to make a bond right here by losing a single water molecule. Make a bond, lose a water molecule. Versus if you were breaking down that molecule and you wanted to get rid of this bond, you wanted two separate nucleotide monomers again, you would add in a single water molecule to break the bond between those two monomers. All right. What do I want to do? Hold on. Guys, I think I'm going to end this so that this is a really short one, and then we're going to pick it up with lipids in the next one, okay? Bye.